Good afternoon, viewers at home. This is another lecture on our educational series of Omega Bio-Edutainment. Um, Today we will continue our discussion on Mendel's law. So this is still under genetics and the environment. I told you earlier on that we are going to do more on genetics, and we actually started with looking at term and terminologies on genetics, and we also started with verification of Mendel's law. And one thing I wanted you to know is just three basic laws of Mendel's. The first law is known as the law of or germinal unit. So this is exactly the law that is actually talking about meiosis, that is a formation of gametes. So by the process called gametogenesis, gameto, that is the coming together of two different gametes. So what this one is actually telling us is that if you have two allele, like this, when you have tall, and this is also another one which is tall. Let's say this one is also tall heterozygously, and this is also. So what first law is actually telling us is that this one will behave as a unit. This one is going to take part during the process of, and we also started with the use of Punnett square, whereby when you now, this is what you have. I told you that you are going to write the gamete on top. This one is female and this one is male, whereas this one is standing for female gamete. So let's say what you have, you are crossing it with this. So this is another T, you are crossing it with this. So by the time you marry this together, it's going to be like this. Marry this with this, it's going to give you this. Marry this with this, this is heterozygous. Marry this with this, is going to be recessive. So, and don't forget that I told you that the Punnett square, you have to interpret. If you do not interpret, you are going to get you are going to be scored low. So this is going to be homozygous tall. This is heterozygous, also what? Tall. This is heterozygous, also what? Heterozygous tall. Recessive, which is going to be short. Recessive, which is going to be so this is just the verification of the first law of Mendel, which is regarded as the law of germinal unit. And actually what that one is actually telling us is that when you closely tall, when you cross it with another parent that is homozygous recessive, so in the first filial generation, you are going to see that all of them, will be tall. All will be tall. And I told you that you will not get the second filial generation. That means you are going to cross capital T, small t, that is heterozygous. You cross it together so that what you are going to have is a capital. Then you are going to have this, as well as this. So when you look at the genotypic ratio, the genotype 1:1, that these two is standing for heterozygously tall, whereas this one is standing for the recessive gene, which is short. So that is, this one is genotypic. But phenotypically, that is physically, what we are going to see is that it is going to be 3:1, 3:1, and that is the verification of Mendel's first law. So then don't forget that this one is parental gamete, so which is represented by P1. So during the law of germinal units, this one will separate. That's what they what he means by segregate. This one is going to separate. That's he means by segregate. This one is going to separate. That is, one of these is going to be donated by the parents, donated by the second parent. So it's going to serve as a germinal unit. That is a single unit. It's going to behave. Although this character, the Mendel character, don't forget that we said traits always exist in pairs. So it always appear in pairs, but each of them is each parent. So that is the first law of Mendel, and that is how you can use Punnett square to do. So if you use Punnett square for this, let's say you use Punnett square as it ought to be, so that your F1 table is going to look like this. Okay, this is the gamete. So you are crossing this with this, so that you have this, you have this, you have this, you have this, so that all this is F1. And don't forget that I told you that F1, we are going to get F2 from crossing F1 and F1 together. So that is, for you to get F2, we are crossing homozygous tall plants or homozygous tall parents. You are crossing them together. So you are crossing two of these. Sorry. One will come here, each of them will come like this. So that by the time you do the crossing, this is going to be homo, this is going to be hetero, this is going to be hetero, this is going to be recessive. This is tall but homo. This is tall but hetero. Okay? This is tall and hetero, whereas this one is recessive. So that is where we get the phenotypic ratio to be 3:1, whereas the genotypic ratio is going to be 1:2:1. Now, Mendel actually made use of the garden pea plants for his experiments, and we are going to see what are the reasons for Mendel to have chosen the garden pea plant over any other plant. So number one, before we go to second law and third law, so we must take note of this. That is, the first law is actually dealing with the germinal unit. This is exactly what was later discovered as the unit of inheritance. Mendel called it unit of inheritance, and that is what we call gene till today. And that is one of the reasons why the father of genetics till today is still Gregor Mendel. Historically, Mendel happened to have come from a very poor family, very poor background, that he knew nobody, and the only thing is that he had that passion to study science. He had the passion to study science, and even when he was in University of London, he chose to be he chose to be a monk. He chose to be a monk, and that was where he lived life. It was during this period that he made use of. I think he requested for some pieces, acres of land, for him to be able to study garden pea, and it was he grew a lot of experiments, performed a lot of experiments on this one for his for the verification of his idea. So the first thing is that let's consider why did Mendel chose garden pea plants. The first one is that many varieties of garden pea plants are available. They are available and they are also observable. So, because their traits, many varieties, a lot of varieties are available, and that it has a lot of traits or characteristics that are heritable. That is, then that is number one. Number two is just that this garden pea plant can be self-pollinated. It can pollination. That is, it might not actually need the influence of an external factor for pollination. That is, it might not actually undergo insect pollination, because these plants can easily undergo self-pollination. Then number three, these garden pea plants are easily available. They are easily available. You can easily get them around, easily available. Okay, and apart from being easily available, is that within a very short period of time, it can undergo different, like 3-4 generations within a very short period of time. So for example, now if you are choosing annual crop now, if you are choosing annual crop, some of them will take like 5 years before you'll be able to see the first filial generation. Then it's going to take like another 5 years before it's going to see another, which is second filial generation to third generation. But in garden pea plants, apart from the fact that it is easily available, it can also the life cycle is very easy. Life cycle is very easy to propagate. So because its life cycle is very short, not only short, it also produces large amounts. It also produces large amounts. So whenever you have one, let's say you have 2-3 seed now, by the time it undergoes self-pollination for the first filial generation, what you just discover is that it has propagated itself in large number. So then apart from that is that it has, I said, it has different type of observable traits. Observable traits. What are these traits that Mendel was actually able to observe, and that was one of the reasons he focused. Number one, the seed. If you pick only the seed is either can come out either round. The seed can come out either round, or it can be wrinkled. So the round is going to be dominant. The seed will come out either round or wrinkled. So that this is going to be the allele. Wrinkled seed is going to be an allele to round. If R is representing roundedness. For example, if capital R is representing roundedness, so small r, which is going to be the alternative, is going to represent wrinkleness. So that one is one of the observable. Then apart from that, is under seed alone, you can divide it into two. The form is either round or wrinkled. Then the second one, which is the cotyledon. The cotyledon is either it is yellow. The cotyledon could be that it could be yellow. So that is telling us that yellow is going to be dominant. Yellow or green. And this one is represented by capital letter Y, and this one is represented by small letter y. We are going to see all this as we go on. Then don't forget that I said that traits exist in pairs because we inherit each of these from each parent. Now, apart from the seed, which is divided into the form and cotyledon, now if you still come under the flower of garden pea plant, you have the color of the flower. It is either it is white or it is purple. It is either it is white or purple. So white is going to be dominant all over purple. So that if this one is capital letter or capital letter in heterozygous form, so the the purple is going to be small letter. So you need to know. So for flower of garden pea plants, the color is either white or it can be purple. Now, apart from that, the pod is also now divided into two. The pod in the form can be full or constricted. So full is going to be dominant. And constricted is going to be what? It's going to be, so if this is capital letter F, this is going to be small letter f, that is constricted. Then the color of the pod. The color of the pod is either yellow. It is either yellow or it is also green. It is either yellow or it is also green. Okay. So I told you we have considered three different characteristics of garden pea plants. The fourth one is the seed. Under the seed, the form of the seed can either be round or wrinkled. Then the cotyledon of the seed can either be yellow or it can be green. Again, if you consider the flower, only the color of the flower, it is either it is white or it is purple. Then when you go to the pod, there are two. The form is either you are considering the form or you are considering the color. If you are considering the form, it is full, which is represented by capital letter F, or it is constricted, which is represented by. You now have the color. The color of the pod can either be yellow or green. So still going to be capital letter Y and small letter y as well, just like you see. So that is the fourth character. Now let's go to the stem. The stem. We have considered the characteristics of the seed, that of the flower, and the pod. So let's go to the seed as the stem of the garden pea plant as it was observed and used by Gregor Mendel. So the stem now, so the stem, there are two also forms. There are two forms. The first one is that the placement of the stem, so it can be placed, then it can be the size. You observe the size. So if you are talking about the place, so it is axial or axial pod, or the axial pod is going to be dominant over the green stem. Axial is going to be the place, that is the location of the stem. Is either it is located with axial pod, or it is located with axial, green. So axial is green. Then when you look at the size, it is either it is long. If it is long, that means it is between 6 feet and 7 feet, a bit taller than the size of a human. And the size could be also short if it is less than 1 feet. If it is less than 1 feet. These are typical characteristics of the Mendel's plant. His choice of choosing garden pea plant, and all this is part of what actually made him to observe 3:1 with what we have seen so far. So the F1 generation of all Mendelian plants will give us this ratio, that is all will be heterozygous. All the F1 will be heterozygous. Then the F2, we are going to come with 3:1, and that is the first law of Mendel, Gregor Mendel. So let's try and consider a typical example of this. Let me consider a typical example of this. Okay. Now consider let's consider a typical example, so that we will now see whether it is Mendelian compliant or it is non-compliant with Mendel principles. Okay. So the question says, consider a cross. Don't forget that in genetics, I told you that in genetics is like mathematics. If you cannot interpret the question correctly, you might have a problem. So that is the more you need to understand and get the question correctly first. So the question says, consider a cross between two heterozygous pea plants for a single trait. Let's seed color. Don't forget that the seed color we observed the other time, it is that the seed color could be either yellow or green. The seed could either be yellow or green. Okay. If it is going to, so the allele for the seed color are represented by, so I told you that here, let's capital letter Y represents yellow seeds. Okay. And the small letter is green seeds. So the question now says you should consider a cross between these two. Consider a cross between two heterozygous, that is capital letter Y and small letter y. That is heterozygous. If you cannot consider a cross between them, so this is exactly what you are going to have. That means this is going to be the parental P1 generation. P1 generation. Okay. Don't forget that according to first law, so this one is going to separate during gametogenesis. So this one is going to separate like this and like this. So the same way, this one is going to stand like this and it's going to stand like this, so that you have it this way, you have it this way. And what the first law is, one of these allele will be inherited from the two parents. Okay. So if you now use a Punnett square for that, if you use Punnett square for it, so don't forget that this is going to be the gamete, male gamete, and this. So this one, let it be like this. Then this one too, let it be like this. This one, this, then this one, like this. So that by the time you now cross this cross this, you are going to have something like this. If you cross this with this, you are going to have something like this. You are going to have something like this. When you cross it, you are going to have something like this. Okay, so what, sorry, okay, this is going to be capital letter YY. This is okay. This one with this is this one with this is this. So this one is going to be homozygous yellow. This is going to be heterozygous yellow. The same thing with this, hetero yellow. Whereas this one is going to be recessive and it's going to be green. So that when you talk about the, if you talk about the genotypic ratio, is going to be, genotypic ratio is going to be 1, 2, 1. That is the genotype. Then the phenotype is going to be 3:1. 3:1. So it's like the question is actually talking about, don't forget the verification of Mendel first law, that the first, or this is exactly what starts the first law when you cross it with this, that is yellow with green. So the first filial generation is going to be, all will be heterozygously yellow. And when you now cross this one to be heterozygously yellow to get your F2 now, so this is going to be 1, 2, 1 and a 3:D. So this one is a typical Mendelian ratio compliance. Typical Mendel compliance. So, and we have a lot of examples that you can actually think and show. Is going to show us exactly the same thing. So don't forget that you should know in first law, just like I said, you cross the yellow with green, and by the time you cross the yellow, don't forget, according to the first law that believe in the law of segregation, so the yellow is going, the allele for that yellow is going to be in pair. So, and each of these will be represented in the gamete of the parents. So that in the gamete of the parents, if you now cross them together, the whole of fourth filial generation, I told you, all will be yellow, but they will be heterozygously yellow. All will be yellow. And don't forget that when you now cross F2, which is the second filial generation, is like you are crossing heterozygous with heterozygous. So don't forget that this one will be represented in this gamete, which is a parental. This is going to be the offspring. So this one will be represented in another gamete, which is going to behave as a germinal unit. Then the same way this. So when you now use a Punnett square, you discover that this one, genotypically, it is going to obey the Mendel's law, which is a 1:2:1 genotypically, and phenotypically it is a 3:1. Now, that is that about verification of Mendel's first law. The second law now, which is called the law of independent assortment. The law of independent assortment, or what we call the law of dihybrid inheritance. The first law is regarded as law of germinal unit, or what you can call monohybrid inheritance, because mono is actually talking about one monohybrid. So we are considering just one character at the same. That is, if you are considering the stem, location of the stem, so it is either you are considering whether it is a hazel or whether it is a green. So you are picking the length or the location once. So you are not considering the stem location as well as considering the seed. So that's why it is called the law of monohybrid inheritance. So the second law now, the second law is the what is now regarded as the law of dihybrid. So this is considering two characters at the same time. It's considering two characters at the same time. Is either you are considering the seed, the nature of the seed, whether it is round or it is a wrinkle. Whether it is round or wrinkle. By the time you are considering whether it is round or wrinkle, you are also considering the height, whether it is more than 6 feet or it is less than 6 feet. So these are the probability of what you can see in dihybrid inheritance. So it can be round but short. It can be wrinkle, also short. So that is the law. So this one is a round, which is represented by capital letter R. Then the height is a tall, which is represented by capital letter T. Then again, it can be round, R, but short, that is small letter, round, short. This one is wrinkle, which is the alternative form of capital R is small r. So that is why this one is going to be R, whereas round, this one is wrinkled, and tall, that is capital letter. Whereas this one is also wrinkled, then short as well. So that is the law of dihybrid inheritance. That is what Mendel used for the dihybrid inheritance. And what he observed at the end of the day, and that's why this one is called the law of dihybrid inheritance, or it is called the law of independent assortment. So what the law says is just that during the process of gametogenesis, what that one is telling us, that second law is saying, is that the representation, the chance that this one, the round seed, that this one will behave independently of whether it is tall or whether it is short. So that's why they call it independent assortment. That is, it's not going to take part. It doesn't depend on any natural or artificial condition. So it is going to be sorted out independently of one another. The fact that this one is round does not depend on whether it is going to be tall or it's going to be short. The fact that this one is short, it does not depend on, it's not going to affect the shape as well. So that's why they call it dihybrid. So two characters at the same time. So that is why, for example, now this one, let's assume that R represents roundedness. R represents roundedness. And small letter, so small letter R represents what? Wrinkle. Okay, Then small capital letter T is representing whether the plant is going to be tall, and the alternative form of it is going to be small letter t, which is going to be what? Short. So if we now want to represent this now, the genotype for this is going to, and don't forget that I said that these traits will exist in pair. So it is going to be the form of it's going to be, it can be capital letter R, capital letter R, and like this, or it can be this, like this. So so that this one is the same thing. Phenotypically, it is a round seeded plant, but one is a heterozygous. So when you now cross it with a small letter, which is the alternative of small capital letter is small letter, and alternative of this is this. So this one is only, so that at the end of the day, what we are going to have as gametes, as gamete is going to be capital letter this, then like this. You are going to have it that you are crossing it with this, so that the gamete, don't forget how we arrive at this. You pick this, which is this, then this one is this one is this, then this one is this. So that's why all the F1 for second law, the F1 is going to be what? All will be heterozygous, and all will be what? All will be round and tall. All will be round and tall. So which is represented by capital letter R. I said heterozygous. Capital letter R, small letter R, then capital letter T, small letter t. And don't forget that I said you get your F2. This is now going to be the parental genotype is going to be this. So that is going to be the result of F1. The result of F2 is going to be F1 cross F1. So this is a product of F1. The same thing with this one. So that at the end of the day, what you are going to have is going to be R like this, RT. So this will be the gamete RT. Then capital letter R, small t. Then you have small r, capital T, and you have small r, small t. So this is now going to be the second law. So let's try and use a Punnett square for the second law as well, the Punnett square.